Okay, yeah, so I am a new faculty member in nutritional sciences, but again, I've been on campus for a number of years and had the great opportunity to work for many people from this department and around campus, obviously, for the last couple of years. So my lab really focuses on transcriptional regulation, genomic activation, epigenetics, and sort of chromatin architecture as well. And we apply that to a number of different questions. Today, I'm going to tell you a story about vitamin D metabolism in a project we've been working on for the last couple of years and something that's active in the lab. And so we're going to talk about how we were able to discover a new mechanism for how vitamin D was regulated through some genomic clues that I'll show, and then how we were able to then exploit some of these genomic features then to create some tools that we can use to study health and disease. And hopefully, eventually we'll be able to help guide clinical trials and drug discovery in a lot more intelligent fashion. So before I get going, let's see, I'm not on this thing. Okay, before I get into the data, I just want to thank those that help with this project in particular. Former lab member of ours, Nancy Benkuski, did a lot of the wet lab work that you're going to see, along with myself, and Sungmin Lee and Jordan Town have came with me to my lab as well. And I definitely would be remiss without thanking Kathy Krentz in the animal facility here, as well as the sequencing facility. So all the data you're going to see were all generated right here in this building. So, and obviously our funding from the NIH. So if I were to ask you, why do we care about vitamin D, or what's vitamin D for, right? So everybody's going to say the bone, right? You'll probably say skeleton, bone, something along those lines. And so that is true, right? So at the tip of this iceberg, we see that rickets is up here, right? So vitamin D will prevent and cure rickets, right? And this university's made a lot of money off of that observation over the years. But there's a lot more that happens below the surface of this water as well, right? And so vitamin D is also involved in things like infections, anti-inflammatory features and inflammation, right? So it has an association with autoimmunity, type 1 diabetes, multiple sclerosis, allergies, slows the proliferation of cancer, and all sorts of things, right? So this down here is really what we're focused on now moving forward in the future, right? So we're still focused on calcium and phosphate homeostasis, the tip of the iceberg here, but also these things down below. And so just so we're all on the same page and you understand how this system works, so there's sort of two spheres of vitamin D influence, as I like to call it. One is the endocrine system, right, where vitamin D regulates genes in an endocrine fashion. And in this mode, we have vitamin D3 that's taken in through the diet, right, through supplementation or from UV irradiation in the skin. It then makes its way to the liver, where it undergoes a first hydroxylation reaction, and that's a largely unregulated process. And that 25-hydroxy-D is the major circulating form of vitamin D. So when you go to the doctor and they say, let's check your vitamin D levels, that's exactly what they're checking. They're checking that 25-hydroxy-D level. This then makes its way to the kidney, where it's hydroxylated again by this enzyme CYP27B1. So that's going to be one of the stars of the show today. And this enzyme is highly regulated. Okay, so CYP27B1 is highly regulated, and that second hydroxylation reaction results in the 125 dihydroxyvitamin D3, or I'll call it 125D, or I'll call it calcitriol. So all those names mean the active hormonal form of vitamin D. And so this then degraded then by the CYP24A1 enzyme. And so it's really the balance of these two enzymes that determine how much active hormonal vitamin D you have in the body. And so calcitriol then works together with the parathyroid hormone that's made in the parathyroid gland, right, in response to calcium concentrations in the blood, as well as FGF23, which is made in the bone that responds to the phosphate concentrations in the blood. And all of these hormones act in an endocrine fashion back on the kidney to regulate the expression of CYP27B1, which produces that active hormonal form of vitamin D. Okay, so the major source of production of 125 or calcitriol in the circulation happens in the kidney. All right, so there's also this paracrine or integrin process that can also occur in cells outside of the kidney, right? So any cell outside of the kidney does have the machinery, albeit at a lower level. They have expression of the CYP27B1 enzyme, but it's at a far lower level, right? And so here now we're more concerned about the substrate concentrations, but we'll talk about that a little bit later in the talk. But here then, small amounts of 25D can be turned over into calcitriol, and that then can act locally in those cells, right? And so this has been shown to be true in immune cells like the macrophage and the T cell and skin cells and other things like that. So, and we'll go over a little bit of that today. So all of the actions then of vitamin D are genomic, right? So calcitriol activates the vitamin D receptor. This then sits down on DNA and activates transcription. And it does so at enhancers that are sort of near or far away from transcriptional start sites. And the outcome of this then are genes that are involved in calcium and phosphate homeostasis, bile acid metabolism, or other functional responses, right? So if it's an immune cell, perhaps regulating cytokines or something along those levels, that way. Okay, so and we've looked at this over the last decade or so, right? And so looking at target cells, and we've learned lots of different lessons about transcription in general, but also transcription related to vitamin D target genes as well. And so here's just one example lesson learned about multiple upstream distal enhancers. And so this is a gene rank ligand. This gene is made in osteoblasts, and it signals then to osteoclasts to resorb bone. So it's essential for that function. And so here we see a schematic of the gene, and we see the gene is located here. And all of these little dots are enhancers that we or some of our collaborators have identified, right? And so there are many different enhancers that regulate this gene. And then listed below are different transcription factors and treatments, as we can see these other boxes, right? So they're not utilized in all enhancers, but they are found to be present in certain enhancers, right? And so then all of these different enhancers have different jobs. And so we can make genetic mouse knockouts of these, and we can understand then what the resulting phenotype of these mice are, right? So we can determine a role for that enhancer. Each of these enhancers has a different role that helps contribute to rank ligand expression. So other lessons that we learned are, there's many different complex arrangements of transcription factors. In this project, we looked at multipotent mesenchymal stem cell differentiation. These cells can be differentiated into skeletal cells as well as fat cells, right? And so each of these then require different transcription factor complexes that come together. But what we can learn from these data is we can sort of understand sort of the spatial relationship across the genome, and through lots of bioinformatics, we can understand this relationship in these interactions, and then we can sort of look at and define activity of genes based on the arrangement of some of these transcription factors. And we found that it's very predictive to the eventual end point of the cell, right? If it's a fat cell or if it makes its way into a skeletal cell, these are all predicted by the combination of these transcription factors that all come together. And another lesson that we've learned from this transcription, again, not just vitamin D transcription, but transcription in general, is that there's this sort of distal enhancers have a cooperativity and a hierarchy to them as well. And so this example of this project was MMP13. This is a matrix metalloproteinase gene that's involved in extracellular matrix. In this gene, we defined that there were 3 very distinct enhancers at 10, 20, and 30 kilobases away. And so in the cells where this gene is active, we have this sort of aggregation of these enhancers that come together to regulate this gene. If we then just delete the -10 kilobase enhancer, where this happens to be where the vitamin D activity resides, we only lose vitamin D responsiveness to this gene, right? But the gene is able to be transcribed just fine. However, if we remove the 30 kilobase enhancer from this gene, this has the binding for RUNX2, which is a bone-specific transcription factor. We see that the entire gene unravels, right? So there's a hierarchy to these different enhancers and how they can control genes. Okay, so those are all target genes of vitamin D, but how about the enzymes that are critical to vitamin D metabolism itself, right? What can we learn about these? Okay, and so going back a little bit here to look at the different metabolites here. Just to remind you, 25-hydroxy-D is our substrate for our CYP27B1 enzyme. Again, this happens mainly in the kidney for the circulation. And then this creates the 125-dihydroxyvitamin D, or the calcitriol. which is then degraded then by this CYP24A1 enzyme. So it's the balance of these two enzymes that determines how much hormonally active vitamin D is present in the body at any one time. And this then degrades it down to this 124-25, which is a degradation product, but also has some activity, but that's a conversation for another day. And CYP24 also degrades the substrate 25-hydroxy-D. Okay, so what we wanted to do in this project is we wanted to isolate the 125 production in the kidney from that in the immune cells. Okay, and in the immune cells, I will show you some data here that it's not regulated by these endocrine hormones. It's actually only regulated by inflammation. So we want to try to separate these two mechanisms, and then we can focus then on the impact of localized non-renal production, right? So that second sphere of vitamin D influence, and look at inflammatory control, and look in things like these autoimmune diseases and cancers and so forth, right? So one problem, though, in this using traditional sort of genetics and knockouts is that there's no good Cre driver in the kidney that deletes all of CYP27B1, right? So all those Cre drivers, as we know, are kind of leaky, but it takes very little amount of enzyme to satisfy this requirement for the mouse. So what we found, though, is we are going to create this mouse using enhancer knockouts, and I'm going to show you how we were able to do that. And that's going to give us our kidney-specific deletion. And so, again, to drive home the point, right, so in the kidney, right, we have these very complex regulatory network that controls vitamin D metabolism. It controls calcium and phosphate homeostasis. It revolves around these endocrine hormones, right? So FGF23, PTH, and then calcitriol itself, right? That helps that regulates vitamin D metabolism. Not only CYP27B1, like I talked about, but all of these endocrine hormones also regulate CYP24A1. I'll show you some data on that in a moment. In the non-renal cells, so the non-renal target cell, or the NRTC, as I might call it, right? The only thing that makes CYP27B1 out there is inflammation, right? So these endocrine hormones do not regulate CYP27B1. And over the years, there have been a number of deletion models that have been created that have helped sort of tease out these pathways. And I'll show you at the end sort of where the last couple of years worth of research, how we can fill in some of these blanks at the end. All right, so outline for the talk today. So first, I want to go over isolating this vitamin D metabolism in kidney from the other cell types. Then we're going to talk a little bit about some unique mechanisms of activation and repression of CYP27B1. And then finally, we're going to talk about inflammatory cell production of 125 and its significance in disease and what we're doing for that. Okay, so isolating this vitamin D metabolism. So first, we'll start with just some evidence that shows this reciprocal regulation, right? So you can understand how the system works. And so if we take a mouse and if we inject it with PTH, we have an increase of the CYP27B1 gene, and we have a suppression of CYP24A1, right? So this is mimicking a low serum calcium situation, and low serum calcium sensed by the parathyroid glands. The parathyroid glands will then release parathyroid hormone. This will then increase 125 production, and that will then increase genes like calcium channels and calcium transporters that will help then bring more calcium into the blood, right? And so on the flip side, though, this phosphate sensor, right? So when phosphate is high, that makes more FGF23. When FGF23 is increased, that suppresses the production of 125, and then that ultimately will lower phosphate. And so if you inject a mouse with FGF23, you can mimic this situation, and now we have a suppression of CYP27 and we have an induction of CYP24, right? So CYP27 is making the 125, CYP24 is degrading that calcitriol. And finally, then 125 itself regulates its own production and degradation, right? So severely downregulates CYP27 and upregulates CYP24, right? To maintain that balance in the body. Okay, so I'm going to show you some ChIP-seq data and some of the slides, and I just want to show you how we're going to interpret that data, just so it's clear for everybody. We display it a little bit differently than some folks do. We use a lot of these overlaid ChIP-seq tracks. And so here's a schematic where I'm showing sort of gene 1 here, and I'm immunoprecipitating with 2 different proteins, and I'm drugging with 2 different drugs. And so we have our drug data, which is shown in blue, and we have our vehicle data, which is shown in yellow. And so then any place where we have an increase based on the drug, right, we're going to have more blue. Otherwise, we'll see the yellow from the vehicle, right? So we're doing this all in vivo. This is in mice. So there's already hormones going on, right? So there's already PTH in these mice. There's already, calcitriol in these mice. So there's already a baseline. And when we inject them with other hormones, then we're going to see this difference. And so obviously you can call peaks right across this. Everyone's sort of familiar with this functionality. But now what we're able to do, though, with replication and statistics, is then we can sort of determine what's increased and what's decreased, right? So we're looking at differential regulation in these different enhancers, and we can get far more information out of this than just peak calling, right? So that would be a peak call for both, but now we know that is massively increased, and we can assign a fold value and a p-value and all that to this. Okay, so the important part here is that when you see blue, that's an increase. If you see yellow, that's a decrease. Okay, and I'm sorry if you're blue-green colorblind. There's not a lot of colors that work well for this setup. Okay, so when we started to look first at the genomic regulation of CYP27, we wanted to then, so we can see that it's in a very gene-dense region of the genome here. So CYP27 is shown here, going left to right. And so what we found when we did a chip for CREB, underneath PTH treatment, right? So PTH will activate through the PKA pathway, which will then phosphorylate CREB, and then activates it to activate transcription. That's one pathway that it can affect the genome. And so when we looked at that, what we found is that there were some very kidney-specific peaks, and I'll show you some more data here in a moment that will convince you, kidney-specific peaks that were shown in the intronic regions of these two genes that were upstream from the CYP27B1 gene. And so I've highlighted those here in gray. And so what we found is that when we looked at this is a very rapid increase. So if we treat animals with PTH, isolate their kidneys and do the chip assay, within 15 minutes, we already have an enrichment on these kidney-specific promoters here. Now, this is called the M1 promoter. It's in the intronic region of the METAL1 gene, and M21, it's in the intronic region of this METAL21B gene. This activity then peaks at 30 minutes, and then it's right back down to baseline by 60 minutes. So it's a very rapid activity of PTH on this gene in particular. And remember that PTH induces this gene, right? And vitamin D, calcitriol, then inhibits this gene. So we then can treat these mice with calcitriol, and we know that the receptor is the vitamin D receptor for that. And so if we do a chip looking for the vitamin D receptor here and its partner RXR, we can see that it too binds in these tissue-specific enhancers that we've identified. And the vitamin D receptor is actually increased as well in these in response to 125. Okay, so these, and importantly, then there's nothing going on at the promoter of the CYP27B1 gene or in the gene body itself, right? So it's all in these upstream regions. So to convince you of the tissue-specific nature of this, we reanalyzed some single-cell attack-seq data from a previous publication. And I've highlighted here in red these two tracks from proximal convoluted tubule cells. And we can see that through the attack-seq that chromatin is accessible and open only in the proximal convoluted tubule cells. In fact, if you look at other cells from the kidney itself, like collecting duct and distal convoluted tubule and so forth, right, we do not see that these enhancers are open and active. So this is not only kidney specific, but it's extremely cell type specific. And above are all immune cells, right? So again, in immune regulation, these endocrine hormones are not regulating, and sure enough, these enhancers are not present. Okay, so we wanted to actually do the difficult work to prove that they were enhancers of the CYP27B1 gene. So we made some CRISPR knockouts and created some mice out of these. And these were with Kathy in the animal facility here. We generated these CRISPR mice. These were some of the first CRISPR mice made on campus here a number of years ago. But we made a deletion of this M1 enhancer here, where we deleted 324 base pair segment of this intron, and a much larger M21 deletion here of 5.5 kilobases. So we called this one the M1 IKO and the M21 IKO. And so when we made these mice, and then compared them to either the wild type mice or the global CYP27 knockout mouse, we saw that the M1 mouse had a very similar gross phenotype to the full global knockout. And so importantly, these enhancer knockouts are global as well, right? So we knocked out this enhancer in all cells. So it's not specific to the kidney. But we see that the M1 IKO animals look almost identical to the CYP27 knockout mice. The M21 IKO mice, on the other hand, look very much like wild type. And so when we start to drill then into the actual phenotype of the mice, we see that the M1 IKO animals were nearly identical to the CYP27 global knockout mice. So low serum calcium, low phosphate, extremely high PTH levels because they can't respond to PTH, and extremely low levels of FGF23. And so this then has a pretty extreme effect on the bone. In fact, the CYP27 knockout mice and the M1 IKO animals have almost a complete loss of trabecular bone, as shown here in this micro-CT analysis. And you can see the pitting here in the cortical midshaft bone as well. And so their bone mineral density is extremely low in these animals. You can see they're smaller and the bones are much shorter, and that's due to these malformed growth plates at the ends of their bones as well, right? So these guys got some skeletal issues. Okay, so not going to show you all the data that went into defining all of this, but then what we found is that there's this functional code then for kidney CYP27B1 expression. And so these two different enhancers are regulated or are responsible then for the regulation of this gene by PTH or of FGF23, which is the M21 animals, or 125, which is sort of spread across both of these. But not only did we figure this out, but we've also created these animal models now that are that we can use. And so these animals then have either an insensitivity to PTH, insensitivity to FGF23, or the double deleted mouse then is insensitive to all of these endocrine hormones, right? And so this is an extremely valuable mouse because this mouse has no circulating kidney-produced calcitriol in these animals, okay? So they're not without their issues though, right? I told you their skeleton, the bones are small and they're fragile and all these things, but we can rescue all of that material by feeding them a rescue diet that has high calcium and high fat, high phosphate, and a lactose-containing diet. This saves the skeleton and lowers the PTH and returns this animal to wild type. And so here's a bunch of serum parameters of PTH, FGF, expression of the CYP27 and CYP24, as well as these other vitamin D metabolites that are shown here. And we can see that these are all either up or down relative to wild type. However, if we rescue this animal, then we can return everything back to wild type, except for really the expression of CYP27 due to the genetic mutation that we made, as well as the systemic production of 125, right? So it's undetectable in these animals. And so what this animal then represents is an experimental model that has no circulating kidney-produced 125D. And importantly, all non-renal inflammatory regulation of CYP27B1 is unaffected, right? So I showed you that ATAC-seq data where, those regions were not open they weren't accessible and open, but I'll show you some data here in a moment as well. And so by doing so, by exploiting this these genomic enhancers, we were able to create this kidney-specific knockout of CYP27, essentially, right? It's not specifically, right? But it's more of a function specific or endocrine deficient knockout animal that we can then use for further studies. Okay, so now I'm going to shift quick and go to some observations here, looking at the unique mechanisms of activation and repression of this gene, CYP27B1. And so in this then, again, I showed you earlier that, one way that PTH affects the genome is activation of PKA, phosphorylates CREB, right? And then CREB binds to the genome. Well, it doesn't do this alone, as you guys are all well aware, as this is a genetics department, right? So it uses different co-activator molecules as well. And in particular, CREB likes to use CBP, which is a histone acetyltransferase, right? So it has enzymatic activity to help decondense that chromatin structure. And it also uses these CRTC co-activators. And these cofactors are believed to help solidify that complex of CREB, CBP, and CRTC2, helps it bind to DNA to help facilitate transcription. Okay, So there's more to this story, though. And in most cells, there are these salt-inducible kinases. These salt-inducible kinases are in the AMPK family of kinases. They're responsible for things like involved in gluconeogenesis and lipid metabolism as well. And so some collaborators of ours that we're working with, Mark Wine and Michael Manstadt at Massachusetts General Hospital and Harvard, found that when PTH activates the PKA pathway, it also inhibits these salt-inducible kinases. This then releases the salt-inducible kinases, then are not able to then phosphorylate CRTC2 anymore, and it's released and sent into the nucleus. It can then help in facilitating transcription. And so it also then plays a pivotal role in bone remodeling, this whole process. And so MGH and Radius Health together have been developing these drug inhibitors to these salt-inducible kinases. The two that we analyzed with them is this compound YKL-05099 and SK124. The names are pretty irrelevant for what we're talking about, but we had two different salt-inducible kinases. And so they're trying to develop these to promote bone formation in skeletal cells, but not have the bad side effects of PTH, right? So PTH is very anabolic towards bone. Long term, though, PTH will start to destroy bone. And so they want to avoid that. So they want the bone formation without the catabolic effects. And so we have a gene that's highly regulated by PTH and obviously in the kidney. So we wanted to look at these from a mechanistic perspective and say, when you're giving these drugs to people, what's going to happen to the vitamin D metabolism? And so when we inject these into a mouse, so here's the YKL and the SK124 compound. So we see that when we inject them into a mouse that we have an increase of CYP27 and a decrease of the CYP24, right? So it's acting in the exact same manner as PTH, albeit to a little bit lesser extent. And so we then looked at what was going on in the genome, and what we found is that, looking at the three proteins involved here, right? So CREB, CBP, as well as CRTC2. We found that PTH then was increasing CREB, CBP, and CRTC2 as we found before, right? So this, increase of blue. But now we see that when we look at YKL and the SK124, that the YKL also increases the CREB, CBP, and CRTC2 very similarly as PTH. But in contrast, though, the SK124 compound does not increase CREB. So CREB is still present, right? But it's not increased. It's decreasing the amount of CBP on the genome, but it is increasing CRTC2, right? So it's following that same pattern. And so we thought this was a little curious. And so when we look at the actual serum PTH concentrations in these animals, this YKL compound is actually causing the PTH to go up. So it looks like PTH because it's inducing PTH, right? However, this other compound, SK124, is not raising serum PTH levels at all. And so if I were developing drugs, I would be using this SK compound, and that's exactly what they're doing. And they stopped development on this other one based on some of these results. Okay, but we're not only just looking at PTH and CREB, right? So we're also looking at the suppressive mechanism of 125. And the other thing I didn't say is that FGF23, we don't know what the transcription factor is for FGF23 on the genome. That's something that we're looking at as well in our studies, but we don't know what that transcription factor is. But we know that 125 activates the vitamin D receptor, so we can look at that. And so if we then look at these same treatments of, so PTH again, but then 125 and FGF23, looking at CBP, CRTC2, and SRC1, again, we see that PTH increases CBP, CRTC2, like we've seen before. And then we also looked at SRC1. So SRC1 is also a transcriptional coactivator. We've and many others have shown that it's involved in lots of transcription factors, including the vitamin D receptor. And so we see that then this is also, SRC1 is also increased with treatment of 125 on these tissue-specific enhancers. And so what we also see then, if we look at things like H3K9 acetylation, H3K27 acetylation, and RNA polymerase, as we can see this dominance of blue here, right? So this is high activity from PTH, right, for these different tracks. But then what we're also able to see is that when we treat with 125, now we're getting recruitment of this CRTC2, right? So we don't yet understand why we're getting this increase of CRTC2, but is highly reproducible as well. On top of that, then we're also seeing SRC1 as I just showed you before, but now then we also see increases of an activation mark, right? So H3K9 and H3K27 acetylation are sort of active regions of the genome. And we also see the increase of RNA polymerase in some of these same regions as well using 125. So despite the net result being a suppression, we still see that we have this sort of activating signature that's involved in the regulation, in the suppression by 125. And so, traditionally you might think, okay, well, let's bring in things that, condense the chromatin structure or silence the chromatin, or bring in co-repressor molecules or something along those lines. But we don't see that happening in this case. And so we've only also looked at this at a few different time points. We haven't done an elongated time course. So we may see some changes happen in the genome, but those are quite expensive experiments to conduct. So we're still looking into exactly what's going on with this active mechanism. Okay, so from this section, right, so we learned a little bit more about the regulation by PTH through these different coactivators, right? So it also involves these salt-inducible kinases that we can inhibit, right? So we can sort of end around PTH's activities here, and we can just use these inhibitors to stimulate transcription of CYP27B1, right? Which might be an advantage that we want. We also saw that VDR then bound by calcitriol, right, will then have a net negative result on CYP27 through these different enhancers. And it appears to involve CRTC2 and SRC1 and sort of this active mechanism. And finally, again, we don't know what the transcription factor is for FGF23 signaling. That's binding to the genome and activating transcription, but we do see that it involves this SRC1. And again, the net result of this is a suppression as well. Okay. So for the last little section here of my talk, we'll talk a little bit about inflammatory cell production of 125 and its significance in disease. And so for this, then, I talked about this earlier, in the two different spheres of vitamin D influence, but in this paracrine and intracrine system. And I alluded to at that point that it was all about substrate accessibility, right? And so when we looked at the expression, again, of CYP27B1 in all of these cells outside of those proximal convoluted tubule cells, the expression of this gene is 100-fold less than that, right? So every other tissue is about 100-fold less or more than those proximal convoluted tubule cells. So there's very little gene or enzyme being produced in those cells. So it really comes down to substrate entry into these cells. And so that's governed by megalin. So megalin is a glycoprotein of the LDL family of receptors, and it helps shuttle in 25 hydroxy D into the cells. And this 25 hydroxy D is bound to this albumin-like protein called the vitamin D binding protein, or DBP. And so it's not just freely entering in these cells, it has to be shuttled in by this megalin. There's also a co-receptor called tubulin that's involved. And these receptors aren't specific to 25D. There's lots of things being brought in by megalin into the cell. And so as you can imagine, right, so the main place that this is, that 125 is being made is in the kidney, right? So in the kidney, the megalin expression is extremely high, right? So therefore it has ready access to substrate, right, that it needs, right? So 25D easily makes it into those proximal convoluted tubule cells and then activates and is converted then by a CYP27B1 enzyme. In all these other cells outside of the kidneys and these non-renal cells, right, then this megalin expression is extremely low. But what we've learned is that in inflammation, megalin will increase in these non-renal cells, right? So you can imagine that now in inflammation, you have an increase of the ability to take in the substrate of 25D. And remember when I was talking about those Cre drivers in the kidney, right? It takes very little enzyme to convert any substrate that's available. So if 25D can get into these cells, it's easily able to be made into calcitriol, and then that will satisfy the mouse, right? It will satisfy the body and all their skeletal needs pretty rapidly. Okay, so we know that vitamin D has a pretty extensive role in the immune system. And so in innate immunity, immune cells like the macrophage or monocyte will, again, be able to take up 25-hydroxy-D, convert then into this calcitriol. And these small amounts of calcitriol will then regulate genes that are like downregulate pro-inflammatory cytokines like IL-6 and IL-1 beta and TNF alpha. It also upregulates cathelicidin. Cathelicidin helps in pathogen killing as well. And so in adaptive immunity, this same process then can push T cells now down the more preferential anti-inflammatory pathway of Th2 and Treg phenotype and push them away from the pro-inflammatory Th1 and Th17. And it will also upregulate a anti-inflammatory cytokine IL-10 as well. And so again, all of these have been shown in cell models and animal models to be viable pathways. But you may be saying, okay, I pay attention to the Newsmark. I know that clinical trials have not gone well in recent history for vitamin D, and that's true, right? And so I will highlight one of the most recent studies that was a very large study, had about 50,000 human participants in it, ages 50 plus, with a follow-up of about a little over 5 years for most of these patients. And so in this trial, this was the VITAL trial with vitamin D and omega-3, so multifactorial treatment groups, where they had groups of no vitamin D supplementation, 400 IU or 2,000 IU. And so 400 IU is what is recommended currently. And so when you buy a multivitamin, it's going to have 400 IU in it. That's an extremely low dose. Though 2,000 IU is a little bit more robust dose, but still not an incredibly high dose to be giving to people. But nonetheless, so they do this, and the overall conclusions then from this study, if maybe you've heard, maybe you saw the news, but vitamin D, the endpoints were that they were looking at was cardiovascular disease, stroke, or cancer deaths, right? And so the outcome then from this study was that vitamin D has no effect on any of these parameters. And so, boo-hoo, that was terrible. But if you take these and look at these follow-up analyses, what you found then is that actually those that started vitamin D deficient, right? So sufficiency is considered at this moment, 30 nanograms per mil is a vitamin D sufficiency. Deficiency is, extreme deficiency is below 20 nanograms per mil. And if you then group out those patients that were very deficient, what you found then is that there was 12% less death events, all death after supplementation. And that same group had 15% less plaque accumulation after supplementation. And both of these are very statistically significant in these groups. But the biggest effect then was on these autoimmune diseases. Like I was just saying, we had this immune-related effect. And on these autoimmune diseases, then these symptoms were decreased by 22% in a statistically significant manner after supplementation. So the real problem is that these studies are hard to design because there's no experimental model for vitamin D supplementation, right? So in the concentration that we're using, the duration, or the outcomes, right? And all of this, there's no model that has this without systemic calcitriol interference, right? But as I just told you for the last 30 minutes, we now have a model of that, right? And so if we take our M1, M21 double intronic knockout mice, and if we take a look at, if we inject them with lipopolysaccharides or LPS, right? So it's a very heavy inflammatory insult. But we see that then in the deco mice, like the wild type mice, that the levels of CYP27 are increased identically, right? So I told you earlier that the inflammatory pathway was unaffected by these deletions we made in our enhancers, and that is in fact true. And if we look at some T cells, either CD4 positive or CD8 positive T cells that we've isolated, if we activate them, it's the same story there, right? So these T cells are able to be activated in the double intronic knockout animals are the exact same as the wild type in their CYP27B1 expression. Okay, so and we know that then this has a role in some inflammatory diseases, right? So this is a swine model of cardiovascular disease that's improved with vitamin D supplementation. Here we have pigs that were on diets, high-fat diets, without vitamin D or with vitamin D, and this is not our study. This was a study done a couple years ago. And but we see that there's plaque formation here in these vitamin D deficient diets, but in vitamin D sufficient diets, there is little to no plaque accumulation in these pigs on the same high-fat diet. We also know that it is the inflammatory bowel disease is more severe in VDR knockout mice. And so inflammatory bowel disease, one of the models here is this dextran sodium sulfate induced colitis model, and it's far worse at lower concentrations of DSS in the VDR knockout mice. And the same story here with supplementation. If you have mice that are deficient, they get the colitis far worse than those that are sufficient of vitamin D. And so we're currently looking at both of these models. We have a lot of work in the atherosclerosis model using our double intronic knockout mice, although we're not yet to results on those experiments as they've taken some time to come about. And we're about to embark on this inflammatory bowel disease model as well. Okay, so some current and future goals here related to this particular project in vitamin D metabolism, right? So I've told you many times that this endocrine method and or the mechanism for the endocrine regulation and the inflammatory regulation of CYP27 are completely different. And so we are currently then trying to discover that mechanism for the inflammatory regulation of CYP27B1, which again involves many different CRISPR mice that again we're making here with Kathy's help. And looking at the different, at the genome and looking for different binding factors that are involved in the inflammatory pathway there. We are also still working on this salt-inducible kinase project as well, right? So these SYK inhibitors and their mechanism of genomic activation. We're very interested in how and why 125 is inducing CRTC2 to bind to these enhancers. And obviously, I showed you one example, but we've got genome-wide worth of data, and there's lots of different sites that are very interesting in this biology for why this is occurring. And it turns out that calcitriol itself will upregulate the CRTC2 gene itself, which is both of these are very novel observations. We're also then looking at these genome-wide implications and impacts of the SYK inhibition with the CRTC2 activation in kidney cells and actually in several other cell types as well. We are also looking at tissue levels of 25 and 125D, including this newer technique of mass spectrometry imaging to try to figure out exactly which cells and where these are being made. And so we can, we've got great preliminary data on that, where we can see that in some of our animal models that are, in our inflammatory models, we have more production of 125 in the spleen, right, where we have a collection of those immune cells than the wild type mice. And we're looking at those in the disease models and in inflammation. And so then by using this isolation of 125 production in these double intronic mice, then we're looking at atherosclerosis as well as that inflammatory bowel disease. And so the idea here is that eventually some of these data then will allow us to optimize supplementation by using actual experimental models, right, to define this and to help guide those clinical studies and improve those outcomes. And so just my second to last slide here, we go back to this complex regulatory network, right? So, and now we can overlay all the mice that we've made in this project over the last 5 or 6 years. And so we've been able to answer everything almost that goes on in renal vitamin D metabolism. However, you see the gaping hole over here is that in the inflammatory production of CYP27. So we don't yet understand the mechanism behind this, and we can't control it yet, but hopefully we will pretty soon. Okay, so I'd just like to thank those that help with this project, those in my current lab, in my former lab and advisor in biochemistry, our collaborators that help us measure all of these different metabolites, and importantly, obviously, Kathy here at the animal facility and sequencing as well, as it wouldn't be possible to do all this without their help. and our collaborators at Mass Gen, Harvard, and of course our funding from the NIH. With that, I'd like to take any questions that you have. Thank you very much.
Time for adult rescue time, like at an age they reach where it no longer works.
Well, an age that it no longer works. So if you don't rescue these mice, they don't live a full life. So they end up having issues and they have a much shorter lifespan. I will say though, the rescue takes some time. So the skeletal rescue is pretty quickly, right? Or happens pretty quick. But what takes a lot longer is bringing down the parathyroid hormone. That one takes a lot longer for that to come down. And so, for some of these experiments, it can be 3 to 4 months of a rescue for all of those systemic parameters to come down. So a new thing that we have, and we haven't yet tested these mice, but we have a diphtheria toxin induced model where when you inject the mice with diphtheria toxin, it kills the parathyroid cells. So we could hopefully then rescue these animals in like sort of weeks time frame instead of months time frame to accelerate some of these studies. And as you can imagine, that affects long-term studies when we're working with our atherosclerosis models, because those you high fat feed for 3 months or 4 months right before you see those plaque form in some of these models. So hopefully we can accelerate that.
Disease that are being revealed and things that you're doing. I don't know.
Yeah, so I don't know about indigenous populations in particular. I mean, there's a number of SNPs that occur that are associated with disease or with defects in vitamin D metabolism, right? So you can imagine, a question I usually get is, okay, well, how much supplementation should people take? And so it's pretty safe for most people to take, unless you have, you can take thousands and thousands of units of vitamin D and you're fine, unless you have a defect in one of these genes. And so there are associated SNPs in or around and near our enhancers that we've defined that are associated with things like MS as well. So I don't know about indigenous populations. I could probably look at the sort of sub-dissection of the data to see if there's anything that pops up along there, but yeah.
So, thinking about your clinical data with autoimmune patients, right? They're usually also taking already a bunch of different things. They're taking maybe metabolic inhibitors, also maybe whatever. And I guess you kind of get at that. How are you thinking about how vitamin D might be interacting with those treatments that those patients are on, in addition to the disease that they're in?
Right, and that's a huge problem, right? I mean, so that's a huge question and a huge problem, and hopefully we'll be able to get close. I don't have an answer for that, but hopefully we'll be able to get closer at answering that if we can start to control some of these variables. So what we'd like to be able to say is, X serum level of 25D in your blood is going to lead to Y outcome, positive outcome in our animal models. So then we can go back to humans and say, okay, well then what do we need to achieve that vitamin D level in you versus me versus anybody else, right? So, which is going to be a different number, right? So it's then you're into, personalized kind of values. But if we can sort of define that, then we can start to control some of those variables and look at those interactions, I think.
Questions on the Zoom.
Oh,
Yeah.
Okay.
Okay, good. Thank you.
I've answered everyone's questions perfectly. Great.
Not perfect. But you mentioned earlier that you received a lot of money.
Yeah. Yeah, so there's a big building over there, Wharf. That was started by Harry Steenbach. And so vitamin D, Harry Steenbach's, they started using, they derived the pathways of vitamin D, of 25D, and then Hector DeLuca finding the calcitriol, and synthesizing that. So basically, all of those early patents in that process is what made work capable of existing. So the university has made a lot of money off vitamin D for many years. I'm not sure what the current status of them. So there's some analogs. I know Hector, who is still active over in biochemistry, has a number of different patents, and I'm not sure which ones are still active. I'm not sure. But the idea of using vitamin D analogs as a drug is sort of a dead end, because vitamin D analogs, calcitriol analogs. The problem with calcitriol analogs is that it increases your serum calcium too much, right? So they're all hypercalcemic, and so when they're hypercalcemic, tissues that should not be turned into rocks, right? And so then you have a really difficult time. So your kidneys will calcify and all these other things. So there's always been this goal to try to find therapeutics that are non-hypercalcemic. But anything so far over the decades and decades of research in analogs have been different, done sort of in classical ways, have not found analogs that are truly non-hypercalcemic. So a lot of the therapy now goes back then to supplementation. So if you can increase substrate ability, right? So if you can increase 25, like there's a pretty, there's like a slow release 25 hydroxy D drug that's actually doing pretty well. And so that elevates the levels in the blood, and then the body takes care of the rest, right? So it sort of buffers out what it doesn't need. And so that's proving to be a little bit better strategy. But I will say though, in analog development, none of them have used these sort of newer genomic techniques to try to see, are there differential things that we can find in how we're activating the vitamin D receptor or certain genes, and can we exploit that with different analogs, right? So we haven't looked at that. So I don't think it's completely dead yet, but it's been sort of a not so fruitful field lately. Yeah, it's kind of like vitamin C, right? So it sort of washes out pretty quickly. So that's why I was saying that there's a slow release 25D drug, right? So it'll last a lot longer in the blood. And again, going back to the earlier question about genetic mutations, right? So some folks have defects in that pathway that they'll clear it very quickly, and sometimes they won't clear it. And so, those will make an effect as well, but it's pretty quick.
And then I just have a genetics mechanism question, which is very naive. And I've always dealt with human biologists who get to, and then it will give things have a justification for it, because in a rhabdomyolysis it's just I do in that report. But so you have sometimes like it looks like maybe a dozen enhancers and get a wide range of distances. And you have that really nice cartoon showing how the enhancers might fold onto each other, but it's hard for me to envision spatially that you would have a dozen all sort of. And so with your work, what are you thinking about how those enhancers are actually like physically being arranged, and how they're redundant or not redundant? Yeah.
Right. And so, I mean, that's a big question, right? So there's, assays that can get at that, like the high C assays and those chromosome capture assays. But again, you're just looking at signals then that look like they're close to each other. We don't know exactly what that looks like and who's exactly connected to who in that. So no, I mean, I think that's a really good question. I mean, again, the best way to define a lot of these things is to go in and start mutating them or deleting them, right? Look for their activities, and we've done that. That's obviously very difficult to do on a large scale. So you have to pick targets that are quite interesting and important, like that rank ligand gene that I showed. There's like 10 enhancers that we identified and sort of characterized over the years. And so, but that's a lot of hard work to do that. So there's no easy answer for that, unfortunately. Yeah.